So this is the GitHub repository for the PNAS paper that I'm going to talk about. I'm also going to talk about another paper that came out this summer in the Royal Society, Philosophical Transactions B, oddest name ever for a journal. But this is the repository, and this is where kind of all of the data and all of the analysis code for the PNAS paper is available. And there's huge overlap because well, let me start. So I am here because I am here for this is what I mean, it's ugly. Because I am here for Neurohackweek, which I'm very excited about to be here. The real people who should be speaking about this are Dr. Petra Virtes, who's a Medical Research Council scholar, postdoctoral scholar, and originally a physicist, but now works with me in the Department of Psychiatry at the University of Cambridge, and Tim Rittman, who's actually a clinical fellow, so he works full-time as a psychiatrist, but also does cool research in his spare time. So when I was asked at the end of last week to give this talk, I then frantically emailed with them because I was like, I can't get away with conceptual. I need to know exactly what you did. So hopefully I will be able to explain that, but I am simply representing their really amazing work. So we had two papers that came out this summer. The first is the one that Petra and I are co-first authors on. And the other one is Petra's paper. They both come from the Neuroscience in Psychiatry Network dataset, which is a study of 300 participants who've come in for MRI scans. They're between the ages of 14 to 24. And they have multi-parametric mapping structural data, which means we have quantitative maps for a couple of different things, but specifically we use the magnetization transfer map in the PNAS paper. And then we also have a multi-echo functional and EPI sequence for the functional analyses. I've put the DOIs on these slides, and you should go and read these papers and tweet about them and up our altmetric. No, you should go and have a look because they're very, very cool. Actually, the one thing I will say is that this Philosophical Transactions B paper, it's in a special edition that came from a meeting that brought together biologists and MR imagers and things like that to talk about the neural correlates of BOLD, and apparently leaving the meeting was quite a sort of depressing state because everyone was like, nothing is real. But I really like the special edition that's come out that this paper is a part of, but I really like Petra's paper, the bottom one, because I think it actually does support the idea that there is something biological about what we're doing with MRI. So it's a little bit important to know the results of the PNAS paper. This is the first figure, and what you're looking at is, so basically we have 300 participants, and they all have a measure of cortical thickness, and they also all have a measure of intracortical myelination. So myelination measured in the middle of it's 2/3 of the way down through the cortex. And on the top row, you're seeing just the cortical thickness at the intercept with age. So it's sort of extrapolated to what you would have at age 14. And I hope you can see that primary cortices are particularly thin, and association cortices are thicker, and that's just a nice sanity check that we've got a measure that everyone else has seen before. And you see the opposite for magnetization transfer, which is this sort of proxy measure for myelin. So now what you're seeing, and actually that's illustrated down in this panel, that magnetization transfer is inversely correlated with cortical thickness. So thin cortex is particularly well myelinated, and again, that's a nice sort of sanity check. We know that. What you're looking at in E and G are the changes in cortical thickness in each of these different regions, and the changes in magnetization transfer cross-sectionally with age. All right, so these are our MRI results. This was the first sort of step. But we want to know how these relate to gene expression, and the first, sort of probably the most important thing that you should remember for these analyses is that we used a parcellation. And the parcellation that we created is illustrated here. We took the Desikan-Kiliani atlas, which labels all of the gyri in the free surfer average space, FS average space, and then we sub-parcellated those gyri so that there were regions that were no larger than 500 millimeters squared. That was like a totally arbitrary number. It gave us like a pretty reasonable number of regions, but I think what I really like about this method of parcellation is that when you pull out the name of a region, it still is located within that gyrus. So instead of being like random parcel 74, it's part 5, or it's random part 5 of the rostral middle frontal gyrus, for example. So those are our regions. They're all around about the same size. They're mapped back into subject space for the MRI analyses. And then you can download, you just saw this web page, you can download the data for the 6 individual brains from the microarray data. And we got all of the data for all of them. And each brain, so 6 brains, each brain has 5 files, and the 2 that sort of are particularly important for this pipeline that we developed, Petra developed, is the microarray expression file, which contains the normalized expression values, so exactly as Terry was just explaining. The rows are the probes, and the columns are the rows are the probes, and the columns are the samples. And then there's the more information about each sample that comes in the sample or not file, and that's the file that contains the location information for the samples with the native MRI voxel coordinates, which really I'm not sure exactly what you would use those for. I suppose you'd use them if you were working with the imaging data, but it also has the M&I coordinates that's been calculated, and the rows correspond to the columns in the microarray expression file. Okay. And the microarray expression file is about a gigabyte for each. They're CSV files. So I wanted to be able to like show you, because I got all excited about the Jupyter Notebook, which I do use. I just haven't given presentations with it before. So I this morning I just grabbed like the top 100 lines so that I could sort of show you that there are. I'm only showing you 5 rows, the top 5 rows. There are more rows in here, but these are the 947 samples. And in the sample or not, you now have the 900 rows and these various columns that tell you the structure ID, the slab number, the well ID, the slab type. So these are all linking back to what Terry was showing you. The structure action acronym, the structure name, and then you've got your coordinates at the end here. All right, and the magic is that I don't know if Terry mentioned, I might have missed it, but only 2 of the donors have both hemispheres analysed, so the other 4 have just the left hemisphere. So what we did is we flipped all of the hemispheres so that they were all left hemispheres. So we sort of had like 8 left hemispheres for all of the participants. There's this optional step where we actually went in and, like not manually, but sort of with a little piece of code, removed all of the rows that were not cortex, because for our particular hypotheses we were interested in cortex. You don't have to do that. That's up to, that would be a decision that you would make. And then you find the sample with the closest M&I coordinate across all of those 8 hemispheres, figure out the structure name for that region, and then find all of the samples within that structure and average for all of the genes. So within our parcel that we're matching up, so we're taking the centroid of the region, the 308, find the closest, but then also take every sample that's inside that structure and average those.
The 308 wells across all donors?
No. So, sorry, it's not the wells. Let me come back. We're actually...
After removal of, because most of the wells are in like brainstem and some stuff.
Yeah.
It's very sparse.
So there are...
Are you left with?
So like a lot, like there's still, there's still enough, but there are some regions that pull from the same structure, which I think is maybe what you're trying to get at. So in our, does that make sense? So we're not pulling by well ID. We're averaging across the structures. So that means that there'll be
wells across structures.
Sorry?
I think so.
There are multiple wells within a structure. Yeah. Sorry. Is that yeah?
Okay.
Yeah.
But the 308 is that number of wells, or?
The 308 are our parcellations from our FS average parcellation. Okay. So there are 308 regions that we have defined by subparcellating the MRI data, the FS average data.
How many wells are there after the removal of the wells that are not complex?
So I don't know that off the top of my head, but I could find that for you. You do lose a decent amount, but there's still loads. Like there's still a lot. Once you start averaging, so I think I was preempting an answer. Once you start averaging across structure, now you start to find that some of those 308 regions are coming from the same structure. So you actually can have the same numbers going in because you've averaged everything. So the output is a data file that has 308 columns. So those are our different regions in the FS average atlas, and the 20,000 genes, like all of the different genes for all of them. And yeah, this is what it looks like. And just in case you were wondering, because there was a paper that came up that was bouncing around the interwebs recently, that yes, if you do open this CSV file in Excel, it saves a whole bunch of them as dates and it goes all totally crazy. So you have to be like, yes, you have to be careful about that. Petra caught that. It wasn't something that we read the paper and then had a panic attack about. We knew about it, but it is a problem. So you now have this massive file that is 308, it's not as big as the original Allen data, but it's 308 columns, 20,000 rows. And what are you going to do with that? Like, you can pull out your favorite gene. So you could pull out the one row, and actually, I forgot to put it in this presentation, but it's in the paper that if you pull out myelin basic protein across those 308, so that's just one row, and you correlate that with myelin, yeah, it correlates. How cool, like, thank God. So that's a nice sort of little sanity check that's in the PNAS paper. But what partial least squares regression allows you to do is look for combinations of variables that explain variance in each other. Yeah?
Did you use just one probe from each gene?
I don't think so.
Okay.
But I don't remember.
There's 60,000 probes, so.
Yeah, roughly some reduction.
Yeah,
I feel like that's done for, I think.
You have to do it yourself.
You do?
Yeah.
So you can follow like the Allen Steer recommendations, or pick one, max, or average.
There are some like ontological plays. It's an art, I have to say.
Yeah.
Like,
Okay.
good literature, it's an art.
So the microarray expression, let me go back, are the probes and the samples. Okay, so no, we'll have averaged across all of the probes.
Average.
I'm pretty confident about that.
across all the probes?
I think so, yeah. Whatever is in the microarray expression, so there's this like huge averaging. So that was something that when I was like emailing with Petra before giving this presentation, she wanted to make sure that you guys sort of knew that this is the decision that we have made with this pipeline. But there are obviously, as everyone in this room knows, there's lots of decisions that you can make along the way. So I am pretty confident that whatever is in the microarray expression file, we average all of them. If there are multiple probes, we average multiple probes.
If you guys are interested, that's how we spent a lot of time doing different reliability analyses for different ways, and averaging seems to be the best for minimizing all the optimal things you're trying to optimize.
That's good. I would have guessed that's what Petra has done. I know the other thing that she's done is not averaged across different donors. So I know that she's also replicated the analyses by taking samples from individual donors, and they don't, obviously there's like a little bit of difference when you've taken 20,000, but the sort of take-home message from the results tends to be very similar. So there isn't a massive difference in the different brains. And yes, I do think that this, I wasn't there for all of these conversations between Petra and Tim, but I think that they probably went along that same journey that you went on. That averaging keeps a lot of the signal. So I was up to PLS. So we've got, here we go. So what it does is we have our predictor variables, which is our gene matrix, which is 308 regions by 20,000 genes. And then we can have response variables, which they just need to be across those 308 different regions. So in the PNAS paper, the response variables are those 4 results that I showed you in the first figure, which is the cortical thickness, age 14, magnetization transfer, age 14, change in correlation with age, slope in cortical thickness with age and magnetization transfer. So those are our 4. And in the Royal Society paper, the response variables were intramodular degree. So this was using fMRI to create, with graph theory, to create networks, and you can look for the degree within module, the number of connections for each region within a module, the number of connections that go to other modules, and the mean connection distance, the Euclidean connection distance of each node. So in this one, the response variables are 3 columns with 308 different regions. And PLS gives you 2 different outputs. The first is a weighting for each gene that tells you kind of how much the signal for this particular gene is explaining variance. And you can be sort of positive or negative, so you're upregulated or downregulated, and then the ones in the middle are not really contributing anything to the component. And you can also have a score for the region, and that tells you where you find this particular pattern of genes. Okay. I'm like giving you like a whole bunch of methods. What you can do with those gene ontology, with those rankings of genes, is gene ontology analyses, and there are these 2 websites, Go Gene Ontology Gorilla, and Whitaker, that are friends that work together. They link up on the web page. And what you do is a lot of the genes have been linked to certain words, certain terms. There's tons and tons of work that have been done on all of these different genes, and you can basically take these 20,000 genes ordered, so your ranked list of genes, and look to see if there are certain words that will pop up when you have that pattern of genes. Or you can flip it around and you can look at the ones that are downregulated as well. And I'll show you results in just a sec. So this is the results for structure. What you're looking at here are the weightings for the PLS component. Sorry, not the weightings, the scores. The regional scores for PLS component 1 and PLS component 2. You can see that they're independent. They sort of, by definition, they're explaining different types of variance. And what we found is that the first component was explaining variance in the intercept, so explaining the differences in regions by whether they're like primary or association, and they go in opposite directions because we know CT and MT are highly correlated. What's really nice about PLS is it's totally fine to have things that are correlated that go in. They'll pull out as combinations. And then we had a different, and these genes, you tend to find these genes particularly in visual cortex, but also a little bit in primary somatosensory, primary motor, and they're sort of particularly down-regulated in kind of medial prefrontal. It's also got a Y gradient. And then PLS2, which we found more interesting because this was the pattern of genes that was explaining variance in the change with age. And here you see that these are the genes that are particularly, that this pattern of genes are found in lateral prefrontal cortex. So these, where you see this pattern of genes, are the areas that are still continuing to change through adolescence. And then this is, we sort of made this figure as a simplified version for publication of the huge gene ontology tree that comes out when you do gorilla. And what you can see that these are the particular words that are associated with that ranking of genes. And they're related to synaptic transmission, regulation of ion channel glutamate receptor signaling. And what's, just as a side, what's really actually amazing is these didn't have to be even anything to do with the central nervous system, right? These biological processes are absolutely everything. And we're only ranking them according to kind of correlations within the brain. So there's even getting something that is related to the brain is kind of cool. But the fact that we pull out something that we think is a mechanism for how the brain might be still continuing to develop through adolescence is particularly nice. And then the results for the Petra's study in the Royal Society, this is PLS1, this is PLS2, and PLS1 was very highly expressed in primary regions, and PLS2, this is a different visualization, so this is from Rovigo, and the words that she found that were related to the genes that were expressed for PLS2 were highly, so PLS2 was highly correlated with connection distance, so long range connections are related to, regions that have long range connections also have genes that are related to metabolism. So that's why what we like about this paper is the fact that there is a mechanism through which, if you have long-range connections that are synced up by BOLD, you may also have this sort of a lot of metabolism going on in those different regions. So I showed you already. That's where the code is for the PNAS paper. The input data, which is the big 308 by 20,000 genes, that data is available there. So if you wanted to get, and so is the free SOFA parcellation. So if you wanted to get started with that, you can jump right on in. And thank you. Yeah.
I know it's hard because there's not a lot of genes that overlap across the samples, but did you find any correlation with the gene expressions with age that are changing in some of those?
Like as in like individual rows. So we purposefully haven't done that because that takes us into this whole kind of like multiple comparisons nightmare. So we didn't. But an additional analysis that we did do was there are two published lists of genes that have been shown to be, one is related to oligodendrocytes, so there's a sort of list of genes that have been shown to be related to oligodendrocytes, which are the cells that myelinate, and the other list of genes have been shown to be related to schizophrenia. And what we did for both of those gene lists is we looked to see where those genes were in the ranking, and when you do this big ranking, and they are more likely than chance, so more likely than random, to be in the top or the bottom of PLS2 for the development one. So effectively what I'm saying is that if we had taken those, let's say the schizophrenia genes, and correlated them with the amount that each region was changing, we would have shown that, if you do that correlation, you see it, it's there. But we didn't do that because that's sort of an analysis pathway that's a very difficult to decide on what's significant and what's not.
And that's using the gene enrichment.
Sorry?
The gene enrichment analysis stuff.
Is that when we, no, hang on, ask the question again. Okay, yeah, sorry, yeah.
How did you decide on statistical significance of your partially square components?
So they're bootstrapped first, and then just by permutation testing.
Do you do any rotations when you bootstrap?
I don't think so.
I bet you do.
Do you think I do? Okay. It's done in MATLAB.
The sign's invariant, so you'll end up with exact opposites. So you have to do it
But
progressively.
if you do, that causes false positives, that inflates false positives.
But it wouldn't work otherwise.
Yeah, so it's
It does work, but it's just either it goes this way or the other way. Wrong. You can get basically random noise. I'm not saying this is.
Yeah. So hang on. So what? So does anyone do? It's just the like PLS regression command in MATLAB. So if anyone has run that particular command, the input are the two like files that I just described to you, and the output are the PLS components. So there's no additional step on top of that. So that's how you would find out. The code is also in that repository, so we'll be able to answer that. Again, sorry, that's...
I'll look at that, yeah.
Yeah.
It's just like, there's one toolbox out there with this forecast rotation built in, and there's a paper
So the-
showing that.
yeah. So that Randy McIntosh's method uses Procrustes with the
Yeah.
bootstrapping and all
He
that.
also has a paper showing that it inflates the false positive rates.
I mean,
So I
you definitely have to. I mean, maybe you don't maybe necessarily need to do Procrustes, but you do need to do some tests to determine whether or not the sign should be flipped, and then
Yeah.
flip it. And so that's
It's closed source documentation doesn't mention rotations. So
PLS command.
the MATLAB.
MATLAB.
Is it
The
part-
bootstrapping stuff is done by Petra, so that code I can point you to in the repo.
I'm just curious. I'm just kind of surveying how people use PLS and all that. Because you're right, like you have so many variables, like kind of the only thing you can think of.
Yeah.
But it's hard to make sense of, and hard to describe as well in a nice way.
I mean, that's where I think the gene ontology and now, like, I think that's where the gorilla stuff is amazing. Is really, really cool. I think you can get when you look at, so I got very excited when I saw panel D. I was like, woo, that looks really cool. That's exactly what I would want to see. But I think it's only when you actually link it up with these biological processes that are related to those gene expressions, that's when you can actually take home a message, I think, about it.
Did you do the same analysis for the last one, first component as well?
So I know that Petra will have run it, but because it wasn't sort of a direction that we cared about with the paper, I've never seen those results. So, yeah, sorry. But I think the files are in the repo, so you could just put them into Gorilla and do it, and it'd be fine. And then you could have a look. Yeah, I've never looked at those outputs, so yeah. Cool.
Awesome.
Thank you. Yeah.